Good morning. So my name's John Inadomi. I'm the division head of GI, and today's a very special day. We have our annual Wade Vollweiler lecture, Dr. Rick Boland, who's the professor of medicine and chief of GI at Baylor University Medical Center in Dallas. Just a little bit of history about Dr. Vollweiler before I introduce Dr. Boland. Dr. Vollweiler was the first division head of GI here. I'm actually the fourth. A big part of that is because Dr. Boland was the division head for 31 years. I've only been here for 3 years, so I have a bit of ways to go. But Dr. Boland was born in Grand Forks, North Dakota, attended Oberlin College, got his MD from Harvard Medical School, and did his training at MGH. He was recruited to UW in 1949. His research interests were quite broad. There were hepatitis and cirrhosis, celiac disease, malabsorption, all of which were very relevant even today. He became president of 3 major national societies. That's the American Association for the Study of Liver Disease, the Western Association of Physicians, and also the AGA, which is very relevant since Dr. Boland is also a past president of the AGA, the American Gastroenterological Association. In honor of Dr. Boland, we have a annual lectureship in his honor. And this year, for 2013, the Wade Vollweiler Visiting Professor is Dr. Rick Boland. Rick was born in Johnson City, New York. He did his undergraduate work at Notre Dame. He had his medical degree at Yale University. And what I didn't know until last night was he was a medical officer at the Gallup Indian Medical Center in New Mexico, and has quite a few stories if we have time at the very end to go over what he was able to do as a medical officer in the Indian Health Services. He had completed his internal medicine residency and GI fellowship at the University of California, San Francisco, and worked with Young Kim, who was one of the pioneers in colon cancer research. He was on the faculty at UCSF and the VA in San Francisco until he was recruited to the University of Michigan, where he was the chief of the GI section at the Ann Arbor VA and the scientific director of their GI oncology cancer center at the University of Michigan. And he was recruited to UC San Diego to become the chief of the Division of Gastroenterology and the associate director for their clinical research cancer center there. And he was there for 8 years as their leader, and then was recruited to Baylor University Medical Center in Dallas in 2003, where he's a professor of medicine at UT Southwestern, also at Texas A&M Health Sciences Center, and adjunct professor at Baylor University Institute of Biomedical Studies. I love this bio sketch. It says, only Rick could get away with this. It says, publications, 350 or so, literally. And the most amazing thing I looked is that his first publication was in 1973. It was a thesis, MD thesis at Yale. It was familial cancer syndromes. And so there's very few people I know who are actually able to remain so focused in something. And again, you've been focused in familial cancer for 40 years. And again, it's not just, the nice thing about Rick is that you'll see in his portfolio is not just the basic science and the translational science, but very clinical studies. And maybe we can hear about these in his talk today. He's got multiple papers in Nature and Science and Gastroenterology and PNS, a lot of clinically important discoveries in terms of fecal DNA screening for colon cancer, looking at methylation and epigenetics, precision medicine, which we'll hear about today, and also serum markers for colon cancer screening, which I think has a real possibility of being a very disruptive technology for colon cancer screening, and hopefully putting the endoscopists out of business and putting the serum markers on the map. He's had over 18 total NIH grants, 4 of which are active R01s, a bunch of merit reviews, and an impressive list of mentees. And that's probably one of the best markers of real investigators. And it's incredible the number of people he's mentored, including Bob Brassolero, who's the chief of GI of MD Anderson, and John Carruthers, who's now the chief of medicine, or the chair of medicine at the University of Michigan. And in terms of national honors, he was the president of the American Gastroenterological Association in 2011, remains on our leadership cabinet for that national organization. And this week, actually this evening, he'll be talking about managing familial colon cancer at our Pacific Northwest GI Society, if you can manage to go to that, tomorrow morning at 8. He'll be talking about the epigenetics of colorectal cancer at our GI Frontiers. And this morning, he will be talking about where are we headed with precision medicine. So Rick, thanks so much for coming, and we look forward to your talks.
Thank you very much. It's a pleasure to be here, and it's nice to see old friends again. And it's always a little embarrassing to hear these introductions, but in any event, I think the thing is I just hung in there longer than anybody else. I want to talk to you about precision medicine. I think it's where we're going. I'm going to use the example of colorectal cancer, but I think that the paradigm is a good one. I'm going to give you some history of how we figured out what colon cancer is all about, and then tell you we're going to get deep into the genomes of colon cancer, which will be a little confusing, but I'll bring you back home. Don't give up too early when we get into this. And then we'll get into the weeds in terms of some mathematical models of how we can use the genomic information to actually treat the disease. So let's go way, way back. So in the 18th century, there was actually a sort of a step forward when people figured out the difference between inflammation and cancer. And if you can imagine, without really good microscopics and you don't have any cytokeratins to stain for, it might be a hard thing to do. Well, but early in the 20th century, aneuploidy was linked to cancer, and fairly early on, the focus was taken to the nucleus, and that people figured out that the cancer was somehow involved with whatever was going on in the nucleus. And then it was in 1910 when the viral hypothesis came up. Now, that idea has come and gone. I'm not going to talk any about it, but it was interesting how far, that's over 100 years old, the concept of the Ralph sarcoma virus. But really, it was in the mid-20th century when it became pretty clear that you could take agents that would damage DNA and you could give cancer to animals. And there were a number of other milestones here. There's no sense to get into that a lot. But it was really in the mid-1980s, well, actually in the 70s with oncogenes, and then in the 1980s with tumor suppressor genes, that we began to really understand how colon cancer occurred. The same thing was happening with breast cancer and prostate and all the rest of that. But this is probably one of the best worked out models is how colon cancer works. So it was in the 70s when Bishop and Varmus chopped up DNA from tumors and then had specific linkers that they could transfect them into to 3T3 cells. And they could make those cells grow a little bit more like tumor cells. And when they pulled those sequences out, they realized that these were altered human cellular genes that were able to drive tumor growth. And they were called oncogenes. And Bishop and Varmus had a nice trip to Stockholm to bask in that particular finding. And oncogenes are normal cellular genes, and they're just activated by something that happens, mutation, amplification, overactivity. Now, Bishop and Varmus probably thought that they had it all figured out, but equally important, if not more so, was the issue of tumor suppressor genes. And these are normally acting genes that restrain the growth of tissues and cells and keep cells well differentiated. They're so-called recessively acting in that you inactivate both copies to make that work, whereas these are activated by mutation. Let's keep this in mind. Oncogenes, like the accelerator that's stuck down, activated by mutation. Tumor suppressor genes, inactivated by lots of ways, often by deletion and by gene silencing. So let's just think about, you want to have a process that can either turn on something that drives growth or turn off something that restrains it. Well, I think that the Normandy beach of this whole story came out in 1989. Actually, there was a paper just before it in 88 by Bert Vogelstein in his group. And they took a bunch of cancers and they used markers. And to make a long story short, and to make this simple to understand, these bars represent the 22 autosomes. And the height of the bar means the number of times, or the proportion of tumors in which there was the loss of DNA. So in the mid-80s, we had pretty crude tools. They were just using restriction fragment length polymorphism analysis. But they were able to see that all of the chromosomes had missing DNA. And this was really kind of taking the aneuploidy issue and making it quantitative. And so the term that was used for this loss of DNA was loss of heterozygosity, or LOH is also called allelic imbalance. Most tumors, but not all of them, this is another important point, was that there was some heterogeneity that began to creep into things. And when they had tools to look at each of these arms, so this would be like this is chromosome 1, this is the short arm and the long arm, so here short, long, short, long. So the fractional allelic loss was 20%, so wherever they were looking there was DNA missing all over the place. And a large proportion of the chromosomes were abnormal in certain locations were really hot spots. And those places ended up being where some genes were found that have really stood the test of time. So the 5P gene was the APC gene, and the 17P thing was a P53 gene, and 18Q was the SMAD gene. And so this was really found in the late 80s. And they identified those genes. Some of them were really serendipitous. It turns out 17P is a pretty short stretch of chromosome. It's a little stump. You know, the chromosomes are numbered, in case you don't know this, from 1 to 22 based on their size. So 17 is not a particularly big chromosome, and the centromere is acrocentric, so the 17P is pretty small. And they only knew of a couple of genes on 17P, and one of them was P53, and they sequenced it, and they found one copy was lost, and the other copy was mutated. So they kind of had the two-hit thing nailed with that one. And the other thing, and we should have been worried from the start, because every tumor was different. So then they said, well, what's the sequence in which this occurs? So they had adenomatous polyps, which are the pre-malignant precursors, and then they had bigger adenomas that sometimes had cancer and cancers. And they found out that the APC gene thing happened early, and the RAS gene mutation occurred in the middle, and the P53 loss was the adenoma to carcinoma event. That was how that occurred. But they wisely didn't swallow the Kool-Aid completely. They just said there were alterations. And it's interesting, if you go back and read these papers, that they left the door open for things other than just point mutations and just deletions of these genes. So this was our first pathway that we had for colorectal cancer. And so when this occurred, when this review came out in 1990, we thought, this is it. This is how cancers occur. So the question is there just one pathway to colorectal cancer? Well, I wouldn't have put that slide up there if the answer was yes, because it really isn't. So just as we were beginning to get a good sense of relaxation and satisfaction about the classical, canonical Vogelsteinian pathway, some disruptions came. And actually, Vogelstein was one of the people who did it. And that was microsatellite instability. And literally, people were trying to do confirmatory studies looking for losses of DNA. And they found that there were lots of mutations. And that was the microsatellite instability issue. And that was due to loss of the DNA mismatch repair system. So about 15% of colon cancers, what was going on was they lost one of the housekeeping functions to make the DNA replication be faithful, and they began to accumulate a lot of mutations. And this pathway looked nothing like that other one. And there were about 10 to the 5th mutations in these colon cancers. They were all point mutations, and they were mostly insertion or deletion lesions at repetitive sequences that are called microsatellites. And there were so many of these mutations, we were pretty sure that they couldn't have been driver mutations. And by the way, most of these mutations were located either in introns or between genes. So at least in 1993, we didn't think that was terribly important. We were a little wrong on that, but it was generally right. They weren't really in genes that much. And so this was a unique pathway to tumor development, about 15% of colon cancers. And and also, because of the fact that we've lost this housekeeping function, this process goes a lot faster than the initial Vogelsteinian pathway. And so these are some of the targets. So we don't have APC and RAS and the rest of that. We have these other genes. And it turns out that there are about 40 or 50 genes in our genome that have a coding repetitive sequence. In other words, they've got, like, in the case of the TGF-beta R2 gene, there are 10 A's in a row. And that is a challenge for the DNA polymerase to replicate accurately. So there are these systems that work in place during S phase of replication, and they can tell if there's been either a misincorporation or a deletion. So if the polymerase makes a mistake, as it does about 1 in 1,000 or something like that times, that will be repaired by the DNA mismatch repair system. The DNA is literally too floppy when the Watson-Crick bonding isn't right. So then there are these other genes too, and it turns out these are all really critical genes involved in the regulation of growth. Like this is the TGF-beta, which is really important to stop colonic epithelial cells from growing, the activin receptor, BAX, one of the apoptosis genes, etc. So we kind of said, all right, so there's two pathways, right? Well, didn't take very long, and we found a third pathway, and that was epigenetic instability. And that is caused by excessive amount of DNA methylation at the promoters of genes. And so this is called the CPG island methylator phenotype. So this doesn't involve mutations. It doesn't involve deletions. It involves excessive silencing of genes. And it's a kind of a random process. But when you silence the right combination of tumor suppressor genes, a cancer can develop that way. So just to give you a little bit of background on abnormalities of DNA methylation, the same fellow, Vogelstein, in 1983, together with his postdoc, Andy Feinberg, they published a paper in which they found they took DNA from cancers and compared it to the DNA from the normal tissue. And they used a restriction enzyme that would cut the DNA. So when you cut the DNA in pieces, you get a recognizable pattern on a gel electrophoresis. And they had two enzymes that cut the same restriction sequence, but one was methylation sensitive and the other one wasn't. That is to say, one of them would cut the DNA only if the sequence was not methylated, and the other one could cut it whether it was methylated or not. So when they ran them side by side, if you got more fragments, if the one that was methylation sensitive was cut into more fragments, then you know there was less methylation of DNA. And the methylation occurs at cytosines in the promoters, at CG dinucleotide pairs. Well, they found this at three different genes. They found that the DNA from cancers had less global methylation. And it seemed to be worse with tumor progression, so they had some metastases, and it was worse there. And then later, Feinberg found that there was an 8 to 10% reduction in 5-methylcytosine in the colorectal adenomas, so he reasoned that this process began pretty early, because you could find this in the adenomatous polyps. So that was how it all began. But then in 1986, Baylor, who was in the same place with Vogelstein, began to look at more focused cancer-relevant genes. The genes that Vogelstein and Feinberg were looking at were like the lens crystalline protein and albumin and things like that, which don't have anything to do with cancer. They were just easy to study at the time. And then when Baylor looked at some genes that were relevant to the cancers he was interested in, which I think was medullary thyroid cancer, he found that there was hypermethylation. And if you're just a casual observer to the field, this can be incredibly confusing. And so let me just tell you this is the bottom line here. Colon cancers, if you just take a population of 100 of them, have global hypomethylation. So there's less methylation, and if you measure the 5-methylcytosine, there's less of it. But a subset of colon cancers, maybe 20%, have specific hypermethylation at promoters, and that silences the tumor suppressor gene. So for about 10 years, we were all confused about what this hypo and hypermethylation were all about. So this global hypomethylation is one process that's there, and that really is characteristic of the typical Vogelsteinian pathway, whereas then this 20% have hypermethylation, and that's now called the CPG island methylator phenotype, or CIMP. And there's widespread hypermethylation, and that's how you silence the tumor suppressor genes. And theoretically, you could take a tumor all the way, you could take normal tissue and completely convert it to a cancer without any mutations, without any deletions, just by silencing enough tumor suppressor genes. But it's a minority of the colon cancers. And what happens is that this methylation, these little lollipops, are the promoter, and these CPG sites are concentrated in the promoter area and in the first exon of genes. And when the cell wants to silence gene expression, it methylates those, which compacts the chromatin and keeps transcription factors from getting at it. It's just a normal way to silence genes. And if you have 2 X chromosomes, some of you do, you only need 1 in a cell. You can't have 2. And so you completely inactivate one of the X chromosomes by extensively methylating everything on the 1 X chromosome. That's the Mary Lyon business. So this is a normal physiological process, but it gets run away in some proportion of people with colon cancer, and we don't really understand quite why. or how that happens. And again, we have a whole group of new genes that are targets of that process. So these regulators of cell cycle. And then this is one of the DNA mismatch repair genes. And interestingly, when you have this CpG island methylator phenotype, some proportion of them, about 12% of all colon cancers, methylate this. And now you have a tumor that explained one whole group of those microsatellite unstable tumors. So we had a whole new group of targets to look at. And the hypomethylation, which is the other way around, that leads to chromosomal instability, which is what Vogelstein had been finding in the very beginning. And in fact, if you take diploid tumor cells or near diploid tumor cells and culture them for months with a hypomethylating agent, they become chromosomally unstable, or this CIN, chromosomal instability. So most colon cancers have this, and this leads to the inactivation of ordinarily silenced oncogenes, in part by allowing for rearrangements to take place. And I'll tell you a little bit more about this in a minute. So this is the hypomethylation of DNA, and it turns out that we've got a lot of parasitic DNA forms in our nucleus, like these long interspersed nuclear elements, line 1s. And when there's methylate, the cell doesn't want those to be expressed, and so they're silenced. And that makes for a stable chromosome. So if you then have some process, like in the laboratory we can make this happen, but something happens in some people's colons, and these become demethylated, you can turn on these oncogenes that are ordinarily silenced by that. And I'm going to skip over this one because that'll take too much time. So what we got now is we have three principal pathways to colorectal cancer. One of them is chromosomal instability. That's the classical initial pathway of APC, RAS, P53, etc. The APC mutations and loss of heterozygosity, and P53 has the same thing going on, and there are KRAS mutations. The DNA tends to be hypomethylated, and there's chromosomal instability. There's a second group, maybe about 40% of all colon cancers, where you have this excessive methylation of promoters, completely different from the global hypomethylation. And this will result in gene silencing. This group has BRAF mutations, not KRAS mutations. And then when the MLH1 gene is methylated, you get acquired microsatellite instability. So the microsatellite instability was, some of it was this stuff right here from the CpG island, and the other bunch was Lynch syndrome. Okay, so it's a little bit complicated, but the bottom line is we've got different kinds of pathways with different genetic targets and completely different processes going on to make the cancer occur. So let's get back to this initial. So this was the initial pathway. Lose the APC gene, you become an adenoma. Get a RAS gene mutation, you get a little bit bigger. You get more advanced with certain other losses, and then the P53 loss is the adenoma to carcinoma step. So that's the adenoma event. That's a growth event, and only about 40% of colon cancers will have this, so there are other ways to skin that cat. And then if you get a KRAS mutation, you're more likely to get a big adenoma, viloglandular adenomas, and pedunculated adenomas too. So that explains some of the variability that we see in the premalignant lesions. That's the adenoma to carcinoma event. If you get a RAS mutation with no APC, it's a dead end, interestingly. You need to have the background of an APC mutation, and that's what a hyperplastic polyp is. And then if you get this BRAF mutation and CIMP, you get C-rated adenomas, which tend to occur in the proximal colon, and then they can go on to a cancer, completely bypassing this other pathway. So it was a little bit complicated. So let's take now, that was the first part. That's kind of the background. So the second issue of three that I'm going to talk about this morning is how many mutations are there in a colorectal cancer? And the real issue is, there are a lot of them. The real question is, are there just too many to count? Is it going to be overwhelming? Are we never going to have any use for it? Or is it something that we can comprehend? So there were several papers that came out, and when they came out, I think most of us were kind of incredulous that this was the case. So if you do a lot of sequencing through cancers, what you find is that most of the sequences are normal. So this particular paper looked at 3.2 megabases of coding DNA and found one mutation per megabase of DNA. And then another paper came out there were, and they got about the exact same number. About 189 genes were mutated at significant frequency, and only about 100 genes per tumor were frequently mutated. Another paper, 1.2 mutations per megabase of tumor DNA, and a paper from Vogelstein's group about the same number. So what everybody found was that if you start sequencing through DNA of tumor cells looking for a mutation, you're going to find one mutation that you want to find for every million bases that you sequence. And that kind of was reassuring, because a cancer has to do a lot of pretty sophisticated activities. It's activities we don't want to have happen, but for that to happen, it's got to have a pretty intact metabolic machinery, so it can't have that many mutations and still survive. So there aren't a whole lot of point mutations. So what it means is if we can find them, they might be relevant. They might be important for driving abnormal growth. So this is from the Greenman paper here. 73 out of 210 they didn't do exhaustive sequencing. It was only 2007, which is so last decade. With the amount of sequencing they did, and don't worry, we're getting there. We'll get to the sequencing, everything. A lot of them didn't have very many somatic mutations, and a few had a whole lot. And we couldn't make any sense of that as recently as 2007. Okay, so then the NIH, or the NCI, decided to invest a lot of money to sequence through a lot of tumors. There was a big debate. Some people said this will be a waste of time. Other people said, no, we can make something of this. And so one by one, these papers have been coming out in Nature and Science, in which you get the complete mutational profile of a whole bunch of tumors. So there'll be a colon cancer paper and a breast cancer paper. And what's interesting is this. Our focus on where the tumor came from, was it a breast cancer, was it a colon cancer or prostate cancer, is a little bit misguided. What we really want to know is what mutations are driving that tumor, and they're not the same just because the tumor came from the colon and rectum. So in this particular paper, they took 276 colon cancers and they did a lot of analyses. And they looked at the whole genome in some proportion of them. And here was the general findings that they had. 16% of the colon cancers were hypermutated. So like I was telling you before, you have some tumors that didn't seem to have any mutations and some have a whole lot. So now we can kind of pull them out and say, let's look at them separately. 3/4 of these had microsatellite instability, usually because of MLH1 methylation. So this was the SIMP tumor with methylation of MLH1. Something that was brand new that no one had really expected was that 25% of this, that's 4% of the total, had polymerase epsilon mutations. So there's a proofread. So when the polymerase goes through and starts to copy the DNA during S phase, there's a lagging portion. So the polymerase holoenzyme has a separate protein that's just proofreading. So it sits along the back side of it and it tries to make sure that there aren't any mutations there. And if you mutate that portion, the synthetic part of the enzyme works fine. The proofreading doesn't. And now you got sloppy DNA replication. And if you excluded the hypermutated cancers, which were greatly overrepresented in the proximal colon, colon cancers and rectal cancers had the same mutational profile. So it helped us understand why we had thought for so many years that there was a difference between rectal cancers and colon cancers. And the reason is a small proportion of the colon cancers are these hypermutated ones. They rarely occur in the rectum, and that really skewed everything. There were 24 genes that were repeatedly mutated in colon cancer. And here were our old friends one more time. So the Vogelstein business from the 1980s stood the test of time. APC, KRAS, P53, and a few others. And there were some new mutations that were found as well. Okay, so non-silent, in other words, really relevant protein-altering somatic mutations range from less than 1 mutation per megabase to a whole lot. So if we tried to see colon cancers as one uniform disease process, it was going to be very confusing. But if you can separate them into the hypermutated and the non-hypermutated, now you can start to make some sense and maybe start to think about how to treat them separately. And 77% of the hypermutated ones were this microsatellite instability. That was the process that was discovered in 1993, and it was mostly due to hypermethylated MLH1. And they had this SIMP problem. And 6 of the other non-microsatellite unstable hypermutated ones were this polymerase epsilon. So now we're starting to get some numbers. And we can say we got this group that have microsatellite instability, a group with the polymerase epsilon, and the rest of them have something else. So if we look at this, we have right here we've got the mutation rate. And the number, it's actually prevalence, but these guys got away with it in nature calling it rate. But anyways, this is a log scale right here. And here's 1. That's 1 mutation per million base pairs. That was that number that I was giving you that Greenman and Schloblum and all those other people found. And if you go across here, that's where most of the tumors are. Most of them have this low mutation rate. And then there's this group of 16% that are hypermutated. And they look different. They've got this microsatellite instability, SIMP, etc. And they're completely accounted for by alterations of the MLH1 gene, that's a DNA mismatch repair gene, or the polymerase epsilon, and the others are something else. So then if we separate out the hypermutated from the non-hypermutated, we say, what are the mutations that are present? And look, here's our old friends again, right down here. 81% of the non-hypermutated tumors have APC mutations. Here's P53. Here's KRAS, right up there where we thought they were. And once you've gotten rid of this other group, it becomes more obvious what's driving these tumors. And there are a lot of other mutations that occur, but there's a lot of diversity out here. And we need to be able to integrate all of these mutations in some way if we want to treat colon cancers medically. So basically, we're going to still try to be either preventing colon cancers or getting the polyps out beforehand, or surgically eradicating them. But when people have metastatic disease, we're going to have to understand this part of it if we want to treat them. So there are our good old friends there. And then over here on the non-hypermutated, it's a different group of genes. And with the microsatellite instability, we saw the activin receptor, we see APC, it's mutated in a different way than over here. And then the TGF-beta receptor too. So once we could separate out the hypermutated from non-hypermutated, we said we're really looking at 2 different diseases. So it's almost like you're a pulmonologist and you're saying, we see this thing. We see these opacities on chest x-rays, and some of them were tuberculosis, and others of them were pneumococcal pneumonia, and we wouldn't even begin to think you'd treat them the same way. And it's the same way here. So we have to kind of divide them up based upon what's driving them. And then there's another issue, and that's the copy number variation. So copy number changes are going to be something that occurs in the case of aneuploid tumors. And what's interesting here is we're taking a look at, and I know this might be a little bit confusing, but let me just take you through this part of it, that there's amplification and overexpression of these 2 genes here. It turns out that the IGF-2 gene can drive tumor growth, and embedded in the middle of that gene is microRNA 480G. So these overlap all the time, but if you have an alteration here, you don't have an alteration in the PIK3CA gene. So they are non-overlapping. So the tumor cell had to have some mutation to achieve that something to drive the tumor, but it wouldn't have both. It would have one or the other. And so you can see that by getting the analysis of DNA at a very granular level, you can start to understand what's driving that tumor. And I think this is one of the most important concepts from this sequencing. And that has to do where are the mutations. So let's take 2 genes, the IDH1 gene, which undergoes a very interesting, not loss of function or gain of function, but change of function. When you get this mutation, it changes its activity. And these are activating mutations. So if you take a look at all the mutations that occur in the IDH1 gene, they're all at the same spot right here, at the substrate binding sites. In the case of the PIK3CA, there are a few spots that activate the gene. So in other words, they're very specific. And if you take a look at this gene, the protein where it folds, these are probably all in the same place. And what that means is we should be able to identify a small compound that can inhibit this. This is an activated oncogene. There should be a small compound that can inhibit that, so you can medically stop that tumor from growing. So they're candidate druggable targets. Down here, for tumor suppressor genes, they can be inactivated anywhere. And that's one of the problems with tumor suppressor genes. So they're going to be a lot different kind of a problem to go after. First of all, how do you get an inactivated gene to come back and become functional? That's hard. But they're also inactivated in lots of different places. So a lot of our activity is going to be focused on finding drugs that can inhibit the activated oncogenes, because they tend to be activated always at the same spot. And also, we're going to, if you just look at all of the different mutations that occur, there are going to be markers for more aggressive disease and less aggressive disease. Now, how we're going to use that, I'm not sure. I mean, to know that someone's got a more aggressive disease may not be terribly helpful, but maybe we can link those markers with specific therapies that work. But this kind of thing is fairly easy to do once you do total genome sequencing. Now, this was a paper, it was a summary of all the mutations that were around, and this is the numbers of somatic mutations in different types of cancers, and I found it really interesting. It's probably impossible to read back there. But there are two key points. Look at this. Colon cancers with microsatellite instability had 500 to 1,500 mutations, whereas those without microsatellite instability had 66. So as you can imagine, there's going to be a very different strategy involved in trying to figure out how to stop this tumor. And look at this, lung cancers. Most smokers with lung cancers have 150 mutations, relevant mutations per tumor, whereas if you're a non-smoker, it's 14. Now, those non-smokers are the ones they're non-smokers, they tend to be women, there are some other demographics that go along with it, but they tend to have mutations at the EGF receptor, and there are drugs, Tarceva, that can make that tumor shrink and go away. Now, it always comes back, and I'm going to address that next, but you can make those tumors shrink down, and it's because there aren't that many supportive actors driving the tumor. It's really driven by a small number. And the other thing, too, is the pediatric tumors down here, glioblastomas and the rest of it, 14, 12, 11, and rhabdoma, 4 mutations. If there are only 4 mutations, someday we should be able to have a drug that can make that completely go away. It's not the same thing as trying to figure out how to make one of these monsters stop growing when you have all those different mutations. Okay, so the final thing to discuss is how are we going to treat metastatic cancer? And I'm going to use the euphemism mathematically. And now this is a little bit of a tough paper, but I think it's worth sticking with it. And it deals with this. First of all, there's a lot of genetic heterogeneity in tumors and metastatic lesions. And there's intratumoral heterogeneity. So if you biopsy a primary colon cancer in 4 different places, you don't get exactly the same mutational profile. Problem number 1. Then when they metastasize, one metastasis might have one mutational profile, another one will have another. Then as these metastases grow, they develop more mutational heterogeneity, and we're going to have to figure out how to deal with that. And then one patient is likely to look different from another. So let's take a look at this interesting paper it's a new journal. It's an online open access journal called eLife. And this represented an interesting collaboration between Vogelstein's group, once again, and a very smart computational biologist named Martin Novak. So here's what they did. They were able to get into the existing literature, and they could say, what's the growth rate of a tumor cell, what's the death rate of tumor cells, and what's the mutation rate of tumor cells, and they got all these numbers together. And then the mathematicians were able to then do a series of modelings. They do thousands and thousands of simulations, changing those growth rates and death rates a little bit each time. And then they tried to say, what is it going to take? What kind of impact, what kind of drugs do we need to have to make that tumor stop growing? So they started with variable outcomes in 6 melanoma cases. So they went back repeatedly and got real live data, but then they would do mathematical simulations. So these are 6 patients who had melanomas. They all had known BRAF mutations, and they were treated with a specific BRAF inhibitor, how you say that. Okay, so these new drugs all have weird names. But they're specifically targeted towards the activated BRAF mutation. And eventually we should have them for all these mutated targets. We don't have one for RAS right now, but eventually we will. And so in this particular case, we get a complete response here. So this dotted line is our ability to detect. And in this particular patient, we got a complete response in a pancreatic metastasis. And these were measured sequentially measured imaging. Here's a brain metastasis. You get a complete response. Here we have stable disease. It doesn't grow. We have a partial disease, a partial response where you can just barely find the tumor, etc. So they're using these combinations of observed responses to targeted therapies and using mathematical modeling to try to figure out how we're going to overcome drug resistance, because these new drugs that we have frequently will make the tumor shrink, but they always come back, either because there are pre-existing mutants within the tumor that are resistant to the therapy, or the new mutation evolves during that. And they've taken all that into account by mathematical modeling. Okay, so what they're using are evolutionary dynamics. And the assumptions were, and this is really interesting, the net growth, tumors, colon cancers grow pretty slowly. The net growth rate is 0.01, so it's growing about 1% per day. The average time between divisions was 7 days. The birth rate is about 0.14, but then they modeled it to 0.13 to 0.15 and the rest of that because some tumors are more aggressive than others. The death rate was 0.13. So you see the net growth rate of 0.01 is just the difference between the birth rate and the death rate. So they could simulate various changes here. And then this is the key point right here, is they said the number of mutations that confer resistance. So you've got a BRAF mutation. How many different mutations in that gene could give you resistance to the drug? And they modeled it between 50 and 200, and they assumed that the mutation rate's about 10 to the minus 9th, and that's all measurable and verifiable. And then they said, let's say we've got a second drug that will be good against the tumor. It could either be another drug against the BRAF, or it could be a drug against a different target in the tumor, simulated between 50 and 200. And then the key thing was, they said, what was the likelihood that there could be one mutation that would confer simultaneous resistance to both drugs? And they made that either 0 or 1, because it was pretty unlikely that there'd be that many mutations that would do that. So then they said, what's the probability of treatment failure with combination therapy? And these patients had these different types of tumors. So they had the number of metastases. So these people had widespread metastatic lesions. They then measured the size of each one, got the total burden. And if they had one drug that was reasonably effective, the likelihood of failure was certain every single time with this size, with this amount of tumor burden. And if they had two drugs, it was virtually certain, unless you had the smallest possible tumor burden. And that was assuming that there could be a mutation that would give you resistance to both. But if you had dual therapy, in which there weren't any mutations that could simultaneously confer resistance to both drugs, there was a really good chance of cure. So what we have here are tumor responses to, and so now they're going to say, well, what about, how do we do monotherapy? We've got two drugs or one drug to use against the tumor. And so this is the total, this is the lesion right here, and this is the number of cells. So we got 10 to the 10th, and we've got a drug that we're starting here, in which there are 50 mutations that could confer resistance. And the death rate, they started with a death rate of 0.13. And all they had to do with this drug was to increase the death rate to 0.15. They made a pretty modest change. And what would happen would be, you'd start with this tumor that was, and this dotted line is always what you can detect. So you got a big tumor here, and you got a very small number of cells that are intrinsically resistant to that drug. And you can make the tumor go away, and this is what happens. It comes back every time. And so now they've made the drug a little bit better. So the death rate went from 0.15 to 0.17 to 0.21. And you can see that they can make the tumor go away a little bit longer, but it's coming back, and it's going to come back here. So if they've just got one drug, even if they've got a really powerful drug that increases the death rate from 0.13 to 0.21, it's still coming back. But when they use dual therapy, what you've got is you can kill the tumor, and there's a small number of cells that are around that are intrinsically resistant to the drug, but they've got their own death rate, and they're just going to die off. And in this case, we've got N12 equals 1. So right here, if that number of tumor cells is not trivial, as it is down here, but it's just 100 cells, eventually that treatment will fail, and this is going to come back again. But with dual therapy, right here, you've got a few cells, and what's happening is you can make the tumor go away, but then it comes back again. So in every instance here, we're getting return of the tumor, but under different circumstances, depending upon birth rates, death rates, and how much you can increase the death rate by this new drug. But it's going to be coming back. So then they say, what's the probability of tumor eradication using 2 drugs? And it really depends on what your tumor burden is in the very beginning. And so right here we've got monotherapy, and you can see that monotherapy has a probability of cure of 1 if you've got a 5 millimeter lesion, but as that gets bigger, the likelihood goes down and down. And this really tells you why adjuvant chemotherapy works, because you're dealing with a very minimal tumor burden, and you've got at least a chance of getting that to work. But here we're talking about lesions that you can actually detect and see, and now that changes things. Now, if we're going to give 2-drug therapy, but there's still 1 mutation that can confer resistance to both, you've got some chance to cure a small lesion, but then because of this 1 mutation that confer resistance to both drugs, boom, down you go. But if you've got, if there's no mutation that confers resistance to both drugs, you've got a much better chance of curing, especially if the number of mutations is either 200 or 50. So if the number of mutations that can confer resistance is low, then you can get a better chance at cure. So based on the simulations, they're saying if we've got 2 drugs and there's not 1 mutation that can simultaneously give you resistance to both, you've got a chance to cure this. So then they looked at people who had real tumors. So these are the metastases from a patient with a pancreatic cancer. So these are the shapes and the sizes, and they summed up the whole thing, and they're represented here. So this big lesion is right here. And this is what happens, and this is what really happens with monotherapy, is you can make those little lesions go away with your therapy. But the big one never really goes away. And in any event, with monotherapy, they all come back. With dual therapy, you do a little bit better, but the lesion keeps coming back if there is 1 mutation that can give you resistance to both of these. And with dual therapy, in which there's no mutation that does that, you can make the small tumors go away, but this big one is too big. So we got to figure out a way. You might have to resect that one. If you get rid of that one, that's the one that came back here. All the rest of the tumors could be cured. Down here, this is a colon cancer, and it's the same story right here, in which you've got, as long as you don't have a tumor of some critical size, monotherapy doesn't work. Dual therapy, if there's a mutation that will confer resistance to both, it will eventually come back. But if that number is 0 and you've got 2 drugs that only have to tilt the birth rate, death rate business by a couple %, you can come up with a curative strategy. Okay, so then when they take a look at well, let me go to the next one because I think this is more important. No, this is the one I want to show you. Okay, so here we've got monotherapy, and you got the same thing here. No, let's go to this one right here. So the issue of sequential therapy was then looked at. So the typical way we use sequential therapy now in 2013 is you try 1 drug, the tumor goes away, and then you give the second drug. Well, right here, we see that you've got 1 drug right here, and that fails. And then when you give the second drug, you get with sequential therapy, the tumor always comes back. And some of the tumors were due to the initially resistant cells, and others are due to the cells that became resistant during therapy. And then if you use the drug simultaneously, you get a certain number that fail. You can get rid of these right here, but you still have this simultaneous treatment therapy. And then sequential therapy, when there isn't a drug, or there isn't a mutation that will make both of these resistant, 1 drug will fail, or 1 drug will work, will give you resistance here, but 2 drugs the 2-drug therapy will fail, but simultaneous therapy with no mutation that will give you simultaneous with a smaller number of mutations that confer resistance will give you a very large proportion of having successful therapy. So essentially, this tells you that if you've got 2 drugs and neither one, and they work on different sites, and there's not a mutation that will confer resistance to both, you should use them at the same time, not sequentially. So in any event, targeted therapy knowing what the mutation is by doing sequencing, finding drugs that can target those mutations can provide an approach to cancer treatment with better outcomes and fewer adverse events. We're going to need multiple drugs, but the interesting thing was they did simulations with 3 and 4 and 5 drugs, and you don't need that many drugs, you just need 2. So there was no additional added benefit by adding more drugs. And outcomes depend upon the number of mutations that confer resistance and whether one will give you resistance to both drugs at the same time. But if we have targets against 2 different genes, the likelihood that there's one mutation that will give you both of those is pretty low. So, and simultaneous but not sequential therapy is suggested. So what we have finally gotten to now is by sequencing through tumors, find the mutations that are driving that. If we can get drugs that are reasonably good and they only have to tilt the balance of birth and death by a little bit, we can eventually treat metastatic cancer. So I'll stop there and take any questions. Thank you.
Rick, thanks so much. That's really a nice summary of a lot of work that you others have done. It's interesting. So there's all these types of ways to look at genetic and epigenetic changes. It seems to me you can go through diagnosis, you can get to prognosis, you can treatment. But I guess some of these things they have to be whether mechanistic or not. And certainly for a screening test, you don't worry about if it's a mechanistic defect. As long as it's there, it predicts either you have cancer or you don't. But certainly for treatment, you got to make sure that whatever target you have is a mechanistic in the tumor genesis. So where are we right now? Do you think we're to the point where we can think about treatment, or we should really focus more on diagnosis using precision medicine, just because of the fact that we don't know much about that?
Right. I think right now, where we are right now is the diagnostic part is a little bit ahead of the therapeutic part. So for one thing, we've got a BRAF inhibitor that's pretty good. But that's good news for melanoma, but only about 10 or 15% of colon cancers have a BRAF mutation. So that's a problem. KRAS is one of the most important of the oncogenic drivers of cancer. It's 95% of pancreatic cancers, it's 40% of colon cancers, and we've got no drug that can touch it. It's really unfortunate because there's one spot, the pocket where the ATPase is where the mutations are all the time. And those mutations activate the KRAS gene and make it drive tumorigenesis. And I think several people have put a whole career into trying to find a drug that will inhibit KRAS, and that's failed. But the NCI has just launched a project to find a drug that will inhibit KRAS. And so Harold Varmus said there's no such thing as an undruggable target. But the people who have studied it said that apparently, even though there's a pocket into which the ATPase sits in, they call it a slippery protein, and no one's been able to figure out how to do that. But maybe there's a way of taking the next downstream step. So we need to get good drugs against KRAS, against all of these other oncogenic drivers. We haven't even begun to touch how do we go after the tumor suppressor genes. I think that the tumor suppressor genes simply allow the unbridled activity of oncogenes. So I think most of the therapeutic approaches will be aimed towards blocking oncogenes. I didn't even talk about the issue of immune therapies, which I think are going to be very important too. And I don't know how they play out mathematically. I thought what was interesting about this article by Bozic et al. Was that they used mathematical models to really come to grips in a real kind of way about how much difference do you need to make in terms of the biology of a tumor cell to make it die, and it's not much. We just need to get a much better armamentarium, and we'll probably be sequencing those tumors, trying to debulk as much as possible, and then going after with I'm guessing 2 drugs based upon these mathematical models. Yes?
From the sequencing that's done at this point, you get lots of different kinds of mutations. And for any one group, once you begin to treat, you have a chance of the 3rd or the 4th or the 5th or the 6th clone coming up. And realistically, is 2 going to be enough? And if you use those right at the beginning, you get rid of the backbone. If all the other ones come up in cells that already have 2, then you've got But if you have a set of cells that is resistant or becomes resistant, how do you deal with that?
That's a really good question, and I can only speculate on where we're going to go with this. We know that for CML, if you know that there's one driver that's really critical, and you have Gleevec sitting around, you can make that, you can suppress that for a very long period of time. I don't know if that means forever. I don't know if we can cure, if we can treat and stop. But if we're going to have to put the hypermutated tumors on the sideline and just focus on the other tumors. And what's reassuring is that it's not that there are thousands of tumors. There are thousands of mutations. There are tens of mutations. And what these simulations suggest is that if you can inhibit two critical drivers, that might be enough. So I'm thinking that time will tell. We're going to need empirical data to know where we're going. But most oncologists, I don't know how many oncologists we have in here, but most oncologists have seen tumors shrink away. I mean, everybody's seen that happen. The problem is they always come back. And then when you go back at them with the same drugs, they don't work anymore. So this just helps us deal with that particular issue. But the fact that you can make fairly bulky tumors shrink and go away has been reassuring. And this simulation, I think, gives some reason to think rationally about how you might attack that problem. So I'm somewhat optimistic that once we have a fuller range of drugs, we need to have a KRAS drug, and we need a few others. I think that we're going to be in good shape. Yes?
Very interesting talk. Thank you. I always wonder, were these the initiating events, or do we get tumors we can see in biopsy and look at things that are way downstream in terms of the initiating event for cancer?
Well, that's a good question. We know that in colon cancer that most of the time the initiating event is loss of APC. And the APC gene is what regulates the intracellular concentrations of beta-catenin, which drives the whole Wnt signaling pathway. So when you lose APC, you've turned on Wnt. So interestingly, you could go after the beta-catenin. If you could do that safely, you could conceivably halt the growth of an adenoma. So that would be an early event. I think what ends up driving metastatic cancer are later events. And what these mathematical simulations suggest is you don't necessarily have to stop everything, because the tumors have a certain mutation rate. They've got a certain death rate. There's a certain penalty to be paid for being hypermutable. So their birth rate and their death rates differ by only 1% or so. And so if we can get a drug that tips the balance, then evolutionarily that tumor will go extinct. So it doesn't have to be an early event. It might be something that's very late. And if the other cells are growing slowly, maybe they will involute with time, if you're just tipping the balance of growth and death. But what you're saying is, I think that what you're saying is that if you went after one of those late mutations that's driving the metastasis, maybe there's this whole group of other underlying mutations that keep growing at some rate. And I think that's a real good question. We're not lucky enough to have enough of these big success stories to see what happens over 10 years.